The cellular processes of mitosis and cytokinesis are responsible for the production of the 200 trillion somatic cells that make up the average human body. Mitosis results in two genetically identical daughter cells through the steps of interphase, prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Interphase accounts for approximately 90% of the cell cycle. During interphase, the cell grows and copies its chromosomes in preparation for cell division. The cell produces proteins through RNA synthesis and creates cytoplasmic organelles such as the endoplasmic reticulum and the mitochondria. Interphase takes place over a 12 to 24-hour period. By the end of interphase, chromosomes duplicated during the synthesis phase cannot be seen individually because they have not yet condensed. Chromatin fibers become more tightly coiled, condensing into discrete chromosomes, as the nucleolus disappears from the nucleus. Each duplicated chromosome now appears as two identical sister chromatids joined together at their centromeres. The mitotic spindle begins to form, and the centrosomes then move away from each other, propelled by the lengthening microtubules between them. In prometaphase, the nuclear envelope fragments, and the microtubules extending from each centrosome can now enter the nuclear area. The chromosomes have now become even more condensed, and the microtubules now attach themselves to the kinetochores at the center of each chromosome. During metaphase, the chromosomes convene on the metaphase plate. Metaphase is the longest stage of mitosis, often lasting about 20 minutes out of the 1 to 2 hour period that the total mitotic stage occurs. Anaphase is the shortest stage of mitosis, lasting only several minutes. The two sister chromatids of each pair part, and each chromatid becomes a full chromosome. The two liberated daughter chromosomes begin moving towards opposite ends of the cell as their kinetochore microtubules shorten. The chromosomes move with the centromere, leading towards the opposite end of the cell. The cell elongates, and by the time anaphase is completed, each end of the elongated cell contains identical and complete collections of chromosomes. In telophase, two daughter nuclei now form in each cell. The nuclear envelope then reforms, and the nucleoli begin to reappear within the nucleus. The chromosomes now become less condensed. Mitosis is now complete. A single nucleus is divided into two genetically equivalent nuclei. As actin filaments interact with myosin molecules on the cytoplasmic side of the cell, a cleavage furrow forms, causing the cell to pinch in two.
Meiosis is important in organisms because it produces gametes such as sperm and egg cells. It is split into two stages, meiosis I and meiosis II. In meiosis I, the homologous chromosomes separate, and in meiosis II, the sister chromatids separate, creating two haploid cells.
Each.
Interphase for meiosis is very similar to mitosis. Protein growth takes place in G1. Chromosomes are still in the very long strands and are replicated in the S phase. In prophase I, the centrosomes begin to move to opposite ends of the cell. Homologous chromosomes condense and pair up. This is a phase at which crossing over between genetic material occurs.
At the chiasmata.
Then, the nuclear membrane fragments during prometaphase I, and homologous chromosomes that are attached at their centromere are attached to the spindle by their kinetochores. In metaphase I, homologous chromosomes are lined up on the metaphase plate, with the centrosomes at the opposite poles of the cell. The homologous chromosomes now separate and are pulled to the opposite side of the cell during anaphase I. In telophase I and cytokinesis, two daughter cells are created. The cell may either reform its nuclear envelope or quickly start meiosis II. Before prophase II, chromosomes do not duplicate because each daughter cell has a diploid number of chromosomes, and the end result will be 4 haploid gametes. During metaphase II, the sister chromatids are lined up on the metaphase plate, ready to be pulled to the opposite ends of the cell. This is followed by anaphase II. In anaphase II, the sister chromatid centromeres are separated, and the sister chromosomes are now pulled to the opposite sides of the cell by the spindle. Finally, telophase and cytokinesis occur again. The nuclear membrane reappears, the spindles disappear, and the cleavage furrow produces 4 haploid cells from the 2 diploid cells that were created by meiosis I.
In conclusion, mitosis makes us who we are today.
It keeps us alive.
Cell division through mitosis allows us to grow, develop, and heal our bodies.
When a man and a woman love each other so very much, they get together and they have a sex.
Each parent contributes a gamete which join together to form a diploid zygote. Each gamete is genetically different due to the crossing over during prophase I. This Mitosis and Meiosis production has been brought to you by
David Young
and Spencer Rice.